Hi, I'm Mara Battagin. I'm working at the Roslin Institute in John Hickey's lab. Our group is developing algorithms, software, and strategies for generating and analyzing data for animal and plant breeders and for human genetics. In the following tutorials, I'm going to introduce you to AlphaSim. The example that I'm going to use were prepared as courses for researchers and animal and plant breeders. AlphaSim is software designed to simulate mainly animal and plant breeding programs. It is freely available on our website and works in different operating systems. The program is written in Fortran 95 and is continuously updated. The major strength of AlphaSim is its flexibility, allowing you the option to run a lot of different scenarios. Before moving to the different features of AlphaSim, we briefly want to explain the core of the program. In the first step, AlphaSim simulates a haplotype sequencing and the initial pedigree. Haplotypes are simulated by calling a coalescent simulator, Max, developed by Chan and colleagues. It simulates haplotype sequences according to a specific ancestral population with specified chromosome size, mutation, recombination rate, and effective population size. The pedigree can be provided by the user or simulated internally by specifying parameters such as the number of generations and the number of individuals and parents for each generation. In the second step, AlphaSim drops haplotypes into the first generation of the pedigree. Dropping haplotypes involves recombination events, which are randomly distributed across the genome or localized in hot and cold spots. After that, AlphaSim samples segregating sites to be either SNPs marker or QTN. In the first step, AlphaSim simulates allele substitution effects. The allele substitution effects at each QTN can be sampled from a normal distribution or from a gamma distribution. At user request, dominance effects can be simulated in addition to additive effects. You can find more information about the equations used in the AlphaSim user manual. In the third step, AlphaSim drops haplotype from the first to the second generation and generate breeding values and phenotypes. And finally, it performs selection and it generates the next generations. AlphaSim has really a lot of features. They can be clustered into 7 main categories: flexibility, selection, biotechnology, population, pedigree, genome, traits. For example, you can simulate different kinds of population, not just animal or plants. You can perform genome editing or select for recombination. AlphaSim allows users to define many traits with different heritability and different variants. You can simulate the full genome or just a fraction, changing the number of chromosomes. All these features can be specified in the parameters file. The parameters file is divided in 15 boxes that we are going to explain in details in additional videos. Finally, AlphaSim produces a lot of output files. The files are divided in 3 main folders: simulate data, chromosomes, and selection. In additional video, we are going to explain the main characteristics of each file. In this video, we have briefly described the method and the features of AlphaSim. In the following videos, we are going to explain in details the parameters file and how to run AlphaSim using small example of plant and animal breeding programs. More complex simulation will also be presented.